as the first speaker, we have Dr. Feng Xu. Please come to the podium. So Dr. Feng Xu has, he completed his PhD in Shanghai Institute of Organic Chemistry. Then he moved to the United States, to the University of Vermont, to complete his postdoctoral studies. And finally, he moved to Merck to complete, is that the final destination, hopefully? To work on total synthesis of indole alkaloids. So he's in Merck since 1996. Thank you for being with us.
Thanks, Abel, for your kind introduction, and thanks organizing committee give me the invitation. So today I will talk to you about green bind design for process evolution. So basically we're talking about asymmetric synthesis of fibergloom. So over the 2 days, you guys already heard John's 12 good green chemistry principles. I'm not going to go over those things, but I will talk to you about how the three-step process we applied into our work. First, we need to go by green. Basically, is the route design, which set the foundation for the greenness, how much we can do. And basically, we're talking about a short synthesis, concise bond connection, and maximize the atom economy efficiency. Then we need to go by step for the green chemistry design, green reaction design, which for this audience, I don't have to talk too much. And on top of that, you still can do further and evolve the process and make it much greener, which I'll highlight it here. So this three process can be continuously cycled until we satisfy ourselves. That means we need to set ourselves the bar really high. And basically, at the end of the day, innovation really is a key. That drives us to find out the best fit chemistry and the best fit process. So this is a fibergloom. Basically, is a potent beta-3 agonist, and currently in clinical trials for the treatment of overactive blood. So over the 30 years, and there is only a single class for this drug, and apparently, for the reason I highlighted here, and patients and the physicians are not satisfied. And there's also an option can do the surgical operation, which for those people not choose to do that, and they have to wear the diaper every day, which makes their life very suffered. So this chemistry show you the first discovery route. Basically, it's a racemic synthesis, and this chiral intermediates is isolated through a separation. Through a intermolecular RCM coupling, and we get this intermediate. So hydrogenation, ring closure, cyclization give us the desired compound. And then we do the EDC coupling, get it to the final product. So during the EDC coupling, there is also epimerization, but that's not a big deal. The problem is overall yield is too low. This route is not green at all. The central problem for this synthesis is this stereochemistry in this molecule. But we get a good lead in this step, and about how to control the stereochemistry. It's not enough, it's not perfect, but it's a good lead. So this is our retrosynthetic analysis. What we wanted to do is go through the reductive amination, close the ring, get the stereochemistry set here. Then we're going to use Horner-Emmons reaction and to set up this molecule and connect this bond, which simplifies the problem down to this aminodiol. So what is the problem for this aminodiol? Basically, they're expensive and not available in large quantity. And this is the one route we showed here in the early on how we make this. Basically, it's a three-step synthesis. Take 2 steps to chop off the nitro group, and also take 1 step, chop off this unneeded piece. But this is the cheap antibiotics on the drug, on the market, we can use it. So the yield is low, and apparently it's not green. How not green is, in order to make 50 kilo of this intermediate, we need to start with 1 metric ton material. So on top of that, we also have issue for this reaction. There is some safety concern. So how do we solve this problem? This is a one-pot process we developed to make this ketoester intermediate. Basically, we take this dirt cheap material, run the amide coupling in one pot, and then we do the bulk protection in the same pot. And again, in the same pot, we treat with the base, and intramolecular acyl migration leads us to this compound, which can be isolated in one pot with 90% yield. So with this molecule in hand, we have started to do our enzymatic dynamic kinetic resolution reduction. So under the reaction conditions, both enantiomer epimerized very fast. The desired enantiomer gets reduced by the enzyme to produce the product, while the undesired one continuously epimerized, and eventually everything converted into the desired molecule. So you start with the racemic, you end up with a color compound. This is a very neat reaction. So the initial enzyme we found is the keto reductase 130. It's really good in the lab scale, but when we scale up, we got some issues because the enzyme is not so stable. The issue can be easily resolved by using different enzyme, which is GDH105. Then, when we scale up, see the reaction is give us a great yield, great EE, excellent DR. And so this is the enzymatic DKR reaction, which is really powerful, allow us to set up the stereochemistry. At that time, the traditional chemistry cannot do it. So with the color molecule in hand, basically, we can do the protection and adjust the oxidation status here to the aldehyde, and then we can run the Horner-Emmons reaction. So it is also interesting to notice, during this Horner-Emmons reaction, the initially formed product also do an aldol reaction with the substrate, form this dimer. But the good thing is the aldol product is the back and forth equilibrium. Eventually, if we age the reaction, everything can be funneled down to the desired product. So we get 80% isolated yield through this step. With this in hand, we need to go ahead to hydrogenate this double bond first because we want to do the ring closure. In order to do the ring closure, we need to do a global deprotection. Get to this intermediate, which immediately cyclized to give us this imine salt. The imine salt can then be isolated through this one pot process and in 95% yield and with reasonable purity. So now we're in the stage to run the reduction. Because remember in the medichem chemistry, and the route give us 10-to-1 selectivity. For us, that's definitely not enough. So we've gone through a lot and screening a bunch of the things, but finally we found if we make this group here a little bigger, and then the color induction gets way better. And we get a great conversion and high DR selectivity. So with this information in hand, now we can move on to finish the synthesis. And basically, we do the one-pot hydrogenation, and we add HMNDS into the reaction mixture over the platinum catalyst. In that case, and we get a 96-to-4 selectivity, and while this thing was in situ protected, upon workup, if TMS fall off, the desired product is isolated, and the undesired diastereoisomer gets rejected down to 0.2%, which is really good. And then, remember I said at the beginning, and here, when the discovery guide run the chemistry, they have issue for the epimerization. Now, how do we solve this problem? We come up with a very nice process, and we're running the EDC coupling in the aqueous, which people usually don't do that. And we're also running the aqueous coupling in alcohol, which allow us to directly isolate the product by charging the ammonia hydroxide. So to do that, we also use the smallest atom in this world, which is a proton, to protect this nitrogen by control the pH. So basically, this nitrogen is a lot more basic than the aniline, and I will adjust the pH. The nitrogen gets protected because it's the aqueous system, and the aniline gets selectively coupled to the desired compound. So by doing that, we get the final product really pure, which is 99.8. And this is an example how, I was talking about green by process optimization. Even we know how we want to run the process, you still have a room to optimize your reaction, make the process as green as possible. So this is the overview for the first gen route. Basically, it's 35% overall yield, which is pretty good. And over 12 steps with only isolated 5 intermediates. And we have no single raw material or reagents are expensive. And we have a great end game. As we discussed, we also gained the good knowledge about how to control the stereoselectivity. But can we do it better? Because at the end of the day, the synthesis efficiency is really a reflection of atom economical efficiency as well as how we control the multiple functional group transformation. So I think we can do better. And so this is our second gen route, retrosynthetic analysis. And basically what we wanted to do is we want to do the intramolecular microaddition or 1,4 addition, rather than go through the Hahn-Emmons reaction, which is also atom economically inefficient. So by doing that, and we think we can do the Sonic-Schiller reaction, introduce this nitro group, nitrophenol group, which is simplified down to this Streck product. So this is our first step, how we're going to do. We need to oxidize this pentyl. So the initial proof of concept was achieved by using Swing oxidation. But Swing oxidation is not a good oxidation to scale up, generate lots of gases, need a low temperature control whatsoever. And we spent a quite good amount of the time to try to figure out what is the best way to oxidize this alcohol. But at the end of the day, we found if we use catalytic amount of the tempo with phenyl-iodobenzene diacetate, and then we can get much better conversion and the reproducible results in lab. So what is the problem for this reaction? The first problem we have is the safety. And we know how good the reaction is, and we know what is the best solvent to run this reaction. But we noticed the reaction requires initiation, which means you charge the reagents until a certain point the reaction takes off. Once the reaction started, it's exothermic, and you cannot control on scale up. If we do the scale up of this reaction, I'm not going to go to work on that day. So how do we solve this problem? The issue is the issue is big, but the solution is also very simple. And all we need to do is add a little bit of water, add a little bit of acetic acid. In that case, we have an instantaneous reaction, and we get a very steady reaction rate. And presumably, because we increase the solubility of this phenyl-iodobenzene diacetate. And in that case, this issue is resolved. So this is what I mean, green by design. And we need to optimize the reaction, understand the issue, and address the safety. And so with that information in hand, I'm going to show you how we develop the process much greener and to make this Streck product. Basically, when we finish the oxidation, we have iodobenzene. Take this mixture, we run a Streck reaction, which is a piece of cake in this case. And we cut the aqueous phase away, and we have the organic phase, but we still have iodobenzene. And then we do the bulk protection. The product is isolated as a solid, and the phenyl-iodobenzene is stay in the mother liquor. All we needed to do is take this, bring into the vinegar, and through the acetic acid per acid to oxidize. In that case, this oxidant is precipitated out. You can filter and recycle back to here using step 1. So by doing that, and we will address this greenness issue and also take care of the waste management. So now we converted this compound into the ketone. Now we are in the stage to run the DKR again. In this case, yes, the keto reductase initially give us a very excellent reactivity in terms of the selectivity and the reaction. But we only get a 50% conversion. What are we missing? Because we're missing the epimerization. We only do the kinetic, we only do a resolution rather than the kinetic dynamic equilibrium. So we go back, study the substrate, now we understand, and we need a higher pH, higher temperature, run the epimerization. But the problem is at this time, and the enzyme won't survive. So how do we solve this problem? Our solution is to go with the evolution of the enzyme. So after 4 rounds, evolution enzyme, and we found that this keto reductase 208 is really good. And the cofactor binding site gets modified, bind much more tight. And the stabilization of the cofactor also allow us to dramatically decrease the enzyme loading tenfold. And the keto reductase is exceptionally active and stable, and which means under the high pH and high temperature, which allows this guy to epimerize quickly. So how good it is, and when we scale up to the 150 kg, we get excellent EE, excellent DR, and excellent isolated yield. So with this compounding, then we run the Sonic-Schiller coupling, which is a piece of cake. And we don't have to isolate this guy, and we directly treat with HCl and debug, and we isolate this salt. And treat with the base, as we expected, intramolecular cyclization give us a desired compound. And then we in situ protect the TMS because we know from the first gen we need TMS. And then run the reduction. So looks pretty good, but there's one problem. We want to make it greener because the catalyst loading is a little bit too high than we expect. So we study the reaction, understand a lot better because the impurities, palladium complex like this, really poison the next stage hydrogenation reaction. So the solution to solve this problem is very easy because we found that those kind of impurity is very soluble in the THF. All we needed to do is during the isolation of this HCl salt and wash the cake with THF. By doing that, we control the palladium level down to 150 ppm, and then we subject it to the reaction, and the reaction performs exceptionally well. And the catalyst load gets dramatically cut down. So with this in hand, now I'm ready to show you the final second gen, how this look like. So start from the pentyl, and we run the tempo oxidation, we run the STRAC, we run the bulk protection, and we have a one part through process. We get this intermediate, which is the first isolated solid, and with good yield over 3 steps. And then we run the Grignard addition, get this to the racemic compound, which is also isolated. Then we run enzyme DKR, which is the enzyme has been done, goes through the enzyme evolution. And then we run the Sonic-Schiller coupling, build up this bond, and we do the deprotection, chop this bulk off, get the third isolated intermediate. This has been done in the through process fashion without isolating any intermediates. Then we do the intramolecular 1,4 addition, and we do the hydrogenation. That's also a through process. This became our penultimate and isolated in great purity as well as good yield. And then we run the final step coupling as we expect. So overall, we improved the yield from the first gen 36 or 32 up to the 32% yield. And now we're talking about a 42 overall % yield. And it's much shorter synthesis and with only 4 isolated intermediates. And the synthesis is also cost effective, and it's also atom economical efficiency. I wish I don't have to use this bulk, otherwise it will be protecting free, synthesis. And we also use the enzyme and use the other catalyst like hydrogenation, but so we call it biocatalysis and the chemocatalysis. And the waste management in this route is also managed. So when we're talking about a waste management, what is a way to measure it? So there's one way to measure the waste management and to determine how green the process is to use the PMI, which we call the process mass index. Basically, the process mass index means in order to make 1 kilo, and you need to count everything you charged into the vessel, the starting materials, the reagents, the solvents, the water, everything. And apparently, the lower number, the better. So this graph show you, over the development cost versus the first GMP delivery we did, and how the PMI goes down. And this is the second gen route, which is pretty good. And I hope this is the example, I show you, how to do the green by design, and we need the go green by the route design, go green by a step reaction design, and also go green by the process optimization design. So basically, at the end of the day, I try to deliver the message, innovation is really the key for us. And the innovation will allow us to find out the best fit chemistry and the best fit process. So let me show you the last slide, which is the most important slide for me. And without this big group of people to get involved, working together, I will not be able to show you the results here. And thank you for your attention.
Thank you, Dr. Xu. So, great presentation about bringing back memories about organic synthesis. So I would like to open this to discussion. Any question for Dr. Xu?
Thanks, Wayne. It was a great talk. Could you expand a little bit on the biocatalysis? Where did the enzyme come from? How did you evolve it to that specific state?
Sure,
Please.
and thank you, Jim. I say thank you, John. And so for those enzymes, we have a collaboration with a company called Codexis. And we have one hit at the beginning. Like I said, when we do the screen of their enzyme pool, our in-house enzyme pool, we know the reaction is going to go well. What we're missing is the cofactor binding stabilization. So nowadays, the enzyme, you know much better than me. Nowadays, the enzyme evolution, once you have a lead, even if it's a 1% yield, you can evolve the enzyme to the desired reaction very quickly, after like a couple rounds of the enzyme evolution. That's how we started.
Where did the enzyme come from in the first place?
In the first place, like I said, it's come from the Codexis company, yeah.
Anybody else? Any other question? If not, let's please receive with applause to Dr. Xu.
Okay, thanks.
Yeah, thank you very much for the introduction and also for all of you to wait until the last third day. I'm sure that it's a bit being exhaustive presentation for all of us. Well, first to put you in context, I would like to tell you that, as you can see, my affiliation is at community college. As any community college, we are working with it's a 2 years program. We give an associate degree to our students. So normally we are dealing with freshmen and with sophomore students. So as you can tell, more or less, for the ones that are in the schools that have graduate programs, we have to really deal with the commitment of students. So it's really not easy to be working with a freshman or with a sophomore student to start building our research group. But I've been very lucky to have very excellent students. We are trying to promote in the peer mentor system, where each one trains the next student. So all the results that I'm going to be showing today are they're coming only for community college. We have a great facility, core facility. There is each faculty member doesn't have a lab. We have a core facility we all share the instrumentation and the space. And what I'm going to present you is the product of the last 2 years, more or less, that we've been working. And then my students are here too, so they came to join this event. Hey, so the title of my presentation is Bioremediation of Pollutants of Pharmaceutical from Residual Waters. So that's a problem that is being around for many years, is as old as the pharmaceutical companies. So we can see here, so most of the efforts on bioremediation are also driven to metals, to organic pollutants, dyes, but very little attention has been paid to pharmaceuticals products. So what is happening? So that in most of the developing countries, I'm originally from Peru, and then you can go to the drugstore or any pharmaceutical to buy your, for example, you have an infection, you can just go and get amoxicillin. A lot of amoxicillin if you want. That's totally wrong, but they are not really reinforcing the rule of getting the antibiotics. So people just, stocks a lot of antibiotics, and then once they expire, they're just thrown into the garbage or down the toilet or whatever is the end of these products. So what is happening is that all these pharmaceuticals molecules are being accumulated in water. So eventually, all these antibiotics or analgesics or any other products are going to develop resistance in microbes. So what happens when you start swimming in a river or in the ocean, so that microbe now is going to be resistant, so now that particular antibiotic is not going to work anymore with you. So every single time you will have to develop new antibiotics, then it would be like a never-ending chain. The second one, importance for, or the problem that we're trying to address is that the EPA has taken a big work on the treatment of pharmaceuticals, and they have listed them as top priority pollutants and of emerging concern, because those are really not pollutants that are going to kill you directly, but they have an impact in the environment. Other effects is something that I learned from my student, is that they change behavior in animals. Fish is not sleeping, birds are not sleeping, for example, for caffeine. They change the ecology. Bees are being affected by the presence of caffeine. Mussels are getting stressed by the presence of pharmaceutical products, antibiotics, and caffeine, and triclosan. All these products that are dispersed in the water are causing problems in the ecology. And one of the most important is that we cannot really control the degradation. So we know that the antibiotics and pharmaceutical products, at the very end stage, they're going to degrade into some other particles or molecules, but we don't really know if these pieces or fragments are going to be more or less toxic than the original compounds. Example of that is polyaromatic hydrocarbons, right? So as we know, polyaromatic hydrocarbons are toxic, but not as toxic as the products that are being created once they enter our body. Our liver takes care of that, and then our body, the intention of our body is to digest the PAH, and eventually it will turn it in a more dangerous molecule. So that's something that could definitely happen with antibiotics. Well, and then they increase the oxygen demand, and steal the oxygen from some normal living organisms that are in water. So what are we doing about the remediation of pharmaceutical products? Well, there are many techniques out there. The most common is activated sludge is the one that has the treatment of the wastewaters. But there are other techniques like ionic exchange. We have reverse osmosis, we have microprecipitation, sedimentation, precipitation, electrode deposition, photodegradation. But bioremediation is, as an area of the biotechnology, is using different type of organisms. And it can be divided into two branches. One is bioaccumulation, and the second one is biosorption. Bioaccumulation uses living organisms. As we can see in that particular slide, is that you have a cell that has, let's see how they fit this. A cell that has the original cell on the top, on the left side. So we have no contaminant, and then at the bottom we have with the pollutant inside. So basically what is going to happen with the bacteria, in this case Bacillus, it will take, by different X mechanisms, it will take, or it will absorb the pollutant, in this case the pharmaceutical or the metal, on the surface. But what are the disadvantages? Well, it has, the advantage would be the selectivity, it's highly selective. But the disadvantage is that we have to babysit the bacteria, right? So we have to give the proper temperature, we have to feed the bacteria, we have to have an appropriate steering to give the living conditions for the bacteria. The second approach is biosorption, which is the area that we work on. We use dead biomass. It's the same. We can have dead cells or any agricultural byproduct. So we can see sawdust, well, these curls that are forming the wood. We have corn cobs, which at some point we also study. And then at the bottom we have tires. So the tires are For example, those are old tires, and then, so these have been approaches that have been used in the past to develop new adsorbents. Now here, in this slide, we are showing the mechanisms, how the pollutants are adsorbed on the adsorbents. Right? The pollutants can be attached by ion exchange, precipitation, complexation, right? Mostly for metals. So this slide is showing for metals, but for pharmaceutical products could be mostly interaction by polar interactions or hydrogen bonding. So that's some of the conclusions that we got at the very end. So the advantages for biosorption, just to go really quickly, well, they have a competitive performance. They are very selective because we have proven these adsorbents even in the presence of salt and other pollutants and other substances, and they have a selectivity. So you can have 1,000 sodium ions and then only one lead, and then our adsorbent is going to selectively take the lead and leave the sodium alone. The same has been observed with pharmaceutical products and caffeine, which are the pollutants that we have been studying. And that's definitely make us think that what would be the possible interactions that are being developed between this system, adsorbent and adsorbate. Cost effective because these are all materials that they have no cost attached to it, well, other than the transportation and some pre-treatment that they might need, but basically the cost is very minimal. We can have a pollutant recovery. Maybe might not be the most ideal thing to do, right? Because nobody really wants to recover pharmaceuticals or products that are decomposed or heavy metals that are very toxic. But if we put this system in the place for remediation of gold, silver, or palladium metals, definitely we want to recover those ions. So the mining industries will be very happy to know that their wastewater, we can recover the gold and those precious metals from their wastewaters for profit of the company. There is no sludge generation, so once we have the adsorbent with the pollutant loaded attached to it, so we can regenerate our our adsorbent and in continuous cycles. Or a second approach that we are trying to work now is the generation of biofuels from these materials, because they are lignocellulosic. Well, we know the problems that we have with lignin, but the chemical modification that we are doing, it kind of weakens the structure, as we see from some of the spectroscopic studies, and then we're trying to analyze if that those changes from the chemical modification can maybe encourage and promote the formation of biofuels. So the adsorbents that we specialize is tea. So whenever you go to visit our lab, we're always boiling tea, and then it's always going to be very relaxing and it's a very pleasant smell. So we work with tea bags. So we're working also on a collaboration with an actual tea company, because in New York City they drink a lot of tea. So for some reason everybody has always the bottled drinks with Nestea, Arizona, Brisk, Snapple. All those companies, they definitely have a big impact in New York. So we're trying to propose this company, why don't you give us your garbage, your spent tea, not the raw tea, is the spent tea after you extract the color, the flavor, and all the taste, right? So and then we want to work with the lignocellulosic, what is left. It's easy preparation, a massive collection, so we can just go to this company and then just get their garbage to use it as a substrate for our reaction. And second approach that we have used, second type of adsorbent is the alginate beads. Not only alginate beads, but also chitosan beads, which are hydrogels. These are natural polymers. In the case of alginate comes from seaweed, and chitosan comes from seashells. I remember that yesterday was Phoebe from Louisiana was working with crawfish, the shells of the crawfish. So this is a kind of a very similar technique that we are using, but instead of using the raw the shell, we use the chitosan, which is the main component, which is the one that is responsible for the for the absorption. So all we do to make these hydrogels is we dissolve it in water. They're very highly soluble in water, and then we gellify them in presence of ions. In the case of alginate, it's very stable hydrogel that we form. Is just by the interaction of the carboxylic groups and the OH with the calcium ions. So it resists acids, resists base. There is really no problem. Even we have been using these beads as encapsulating matrices for our other adsorbents. Here is the setup, the experimental setup that we have in the lab. We have fraction collector to run the column experiments. And later on, very recently, starting this year, we're starting working on chemical modification, right? Because at the beginning we noticed that raw materials, the raw tea leaves, spent tea leaves, they had a very high affinity towards metals and then some other pollutants, pharmaceuticals. So we said that, okay, so since we have identified that carboxyl, OH, right, thiol, alcohol, these functional groups are responsible for the adsorption, what about if we add more functional groups to the surface? We use our materials as a platform to start adding more of these functional groups to promote the adsorption. So the treatment that we have to do to the tea leaves, well, since they have chlorophyll and they have some other dyes inside, we have to do a conventional SOx extraction, and then this technique is excellent for teaching purposes. I mean, now that we're in a community college, definitely that extraction is pretty straightforward. The chemical modifications that we were working with carboxylation, thiolation, and sulfonation. So we're incorporating these three functional groups. Well, not all at the same time, but we're carboxylating chamomile, peppermint, and green tea. So we are doing a very easy reaction with sodium periodate, right, which cleaves the diols, and then we reduce it to produce the sulfonate. The second reaction, we have the thiolation. So we just use the property of the ester formation, the Fischer esterification, to produce an ester. But the particular thing that this reaction has is that the thiol is on the other end. So we're basically adding a thiol to every single OH that we have in our adsorbent. Carboxylation. We use citric acid. So citric acid is a triprotic acid. So we form an ester with one of the carboxylic acid that leaves the other carboxylic acid free. So we lose one carboxylic acid to form the ester, but then we get two more. All these reactions are done in water. So there's definitely no organic solvent. There is nothing really, because we work in, well, definitely we have to work in hoods because of the thiol, because we cannot really avoid the smell of the thiol, but any other than that, this reaction is very simple. So the results that we got initially we wanted to see what is the stability, what happened with our adsorbent. So we ran thermogravimetric analysis to see how much we are deteriorating our material. So you can see at the top graph, we have the difference in just raw materials. We have chamomile, green tea, and peppermint. Definitely chamomile is our weakest adsorbent in terms of rigidity, because as we know, chamomile is a flower. So definitely it's not going to be as strong as leaves. Between green tea and peppermint, there is really not much difference because they're very strong adsorbents. At the bottom, you can see that after the chemical modification. So we run peppermint comparing to carboxylated peppermint and thiolated peppermint. Carboxylated peppermint and thiolated peppermint definitely are weaker adsorbents. So they decompose, they react with temperature. Basically, the curve is shifted more to the left, saying that they don't resist, they don't like heat as much as peppermint, a raw peppermint. So something is going on with our adsorbent. So we're not only adding groups, but you also kind of creating some damage to the adsorbent. But that damage does not prevent the adsorbent to have that strength for absorption of the pollutants. So the presence of these functional groups were confirmed by the FTIR. FTIR was also showing some damage some changes in the structure of the adsorbent. But again, the adsorption test definitely demonstrated that these materials still have a high affinity towards pharmaceuticals and heavy metals. We also did a normal potentiometric titration to determine what is the amount of acidic groups, because this thiol, carboxylic, and sulfonic, the sulfonic acid, they have acidic properties. So we just did a very conventional titration to determine the amount of these acidic groups. This is the scanning electron micrographs for our adsorbents. This is the raw material. At the very end, I will show you what is the change of the adsorbent after the chemical modification. But as you can see here, it's very heterogeneous surface. It's normal to see these surfaces in teeth. Right? So we believe that these pores, these channels, these kind of holes that we have in the adsorbent, definitely they house our adsorbent. So it's something that we need to prove, but so far it seems like it, and then we confirm that with EDX experiments later. There's more characterization, just to demonstrate that the carboxylic and the carbon groups are present in our adsorbent. And here are our absorption experiments. So we were trying enrofloxacin. Enrofloxacin is an antibiotic that is used for animals. And the first graph on the left, we have the hydrogels, the alginate and the chitosan, and then we were obtaining up to 20% removal, so definitely nothing that encouraging, but we're still optimizing the process. So we're trying to see how we can maybe maximize the number of functional groups, but definitely it's a good approach that good results that we're getting so far. On the right, we have the differences in absorption with chamomile. We have all the three chemical modifications, carboxylated, and seems like sulfonated has the maximum absorption. So definitely the incorporation of these new functional groups is creating a higher absorption and retention of these pollutants. In the case on the modified tea leaves, we have penicillin. Penicillin gene G is actually used for humans. So we try to divide which one is the best type of adsorbent. Then we wanted to try one antibiotic for animals and one antibiotic for humans. That's why we chose enrofloxacin. There was really nothing specific about the selection of this other than that. We also use marine algae because we were using alginate beads, the hydrogels. It's a totally different effects or adsorption. But we can see that in some cases, some adsorbents are good at low pHs, some other adsorbents are good at medium pHs, slightly acidic, or neutral pH. In the mass effect, well, we are trying to minimize the amount of material that we were using in the process, and we're getting that definitely these materials achieve a saturation. So you give more and more material adsorbent, but at some point you will reach the stability. Here's some of the results with caffeine and penicillin G again with the modified, and we are again getting a better result with modified adsorbent. Later on, we haven't really run these experiments yet, but we have to do a concentration effect to determine the parameters of equilibrium and apply some isotherms to determine the constants that are definitely going to give better insights into the mechanism. Salinity, what happens if we have a spill or these pollutants are in seawater? So we are also trying to explore what would be the effect of these salts on the adsorption. So definitely it has some effect, so it decreases the adsorption, but at least in caffeine, you can see that it doesn't really go to zero. Crowding agent, we put some detergents inside because, again, our water is not going to be pure. We see a decrease in the adsorption, most likely because of the solubilization or for the formation of some prevents the pass of the pollutant to the adsorbent. Just let me go to the continuous flow. And continuous flow, we are trying to, what about if we take the system to a column to make it more kind of engineering application? So this is a very preliminary data that we get. We definitely have to change the flow. Maybe we have to change the amount of adsorbent, but definitely this process can be made automatic to make a more constant cleaning of the solution. Here is a more reasonable curve for for columns. Here we're eliminating copper, and then we were able to remove up to clean 500 milliliters, so of clean water we're obtaining from the copper solution of 100, initial concentration of 100 ppms. That drop that you see at the very end is we just flushing water to prove that it's tightly bound to the column. What is next? So we can recycle the adsorbent, but the best is that the SEM images, this is one of my last slides. You can see the difference on the structure of the adsorbents after the chemical modification. So we have green tea, carboxylated green tea, sulfonated green tea, and thiolated green tea. So these adsorbents have not really lost the power of absorbing the pollutants, but they have changed definitely the structure. So we're hoping that this change, because of the reaction with acids or with these chemicals that are doing the chemical modification, might have some impact in the structure of lignin, and then open up some cellulose and hemicellulose for the production of biofuel. So we are right now on that stage, so we're trying to see what happens with these materials, if these can be potential to produce these biofuels. So conclusions, well, there is the potential of using tea leaves and the chemical modification is definitely a promising area. So we're still working on optimizing. 25 to 40% is the maximum absorption that we're achieving, but we're still trying to optimize the chemical modification. So we can produce, we're looking for another lab in our institution for the production of biofuels. So this work was completed by, those are all the students from BMCC. Most of them are already transferred to a four-year college. So, and then this work was mostly completed by Aaron and Tenzing, and also finished by Katerina and Michelle, who are right here. Well, and the funding, definitely, we're serving minority serving institutions, so most of the money is coming from this type of entities that fund our research. So thank you very much for your attention.
All right, so in the interest of the time, and I think we can ask one question. There you go.
Do you know why the absorption depends on the pH?
The pH, we believe that since we are changing the pH of a solution, you are ionizing the functional groups that are present on the adsorbent. So in some cases it can be negatively charged, positively charged, or neutral. So definitely the type of interaction that you might form between your pollutant and your adsorbent definitely is going to be strongly dependent on the pH. We know that it's the strongest effect that kind of has the domain of the process. Mm-hmm. Okay. Thank you.
Thank you. So next we have a workshop, and it will be presented by Professor Godivan and
from France, and today I will make you a presentation on a novel lignocellulosic hydrolysate detoxification process using pyrochar from anaerobic digestate state toward an application for bioethanol fermentation. So first, what is anaerobic digestion? Anaerobic digestion is a biological process that can convert organic matter through anaerobic microorganism into biogas and digestate. The main characteristic of anaerobic digestion are temperature mesophilic or thermophilic, a pH that can vary from 6.5 to 7.5, and an hydrolytic retention time that vary from 60 to 100 days. So through the anaerobic digestion process, you can produce a biogas, which is mainly composed of methane and carbon dioxide. At farm scale, this biogas is mainly converted through cogeneration heat and power system with an electricity efficiency of 35%. But you also generate some waste called digestate that correspond to the mixture of microbial biomass and indigested material that account for 45 to 75% of the initial organic matter feed. Due to the growing number of anaerobic digestion in Europe, there is now urgent interest on developing some alternative valorization routes for this digested valorization. Among them, here are represented the process of anaerobic digestion. So you have the digestate output. At farm scale, there is generally a mechanical screw separation to obtain a liquid digestate and solid digestate. The common valorization route today of liquid digestate are fertilizers, but there is also today growing interest for algae cultivation due to the high content of nitrogen and potassium present in the liquid digestate phase. For the solid digestate, the main valorization route today is salamanders, but in the literature you can find some new valorization routes which are still at lab scale. Among them, there is pyrolysis, combustion process, biofuel production, mainly bioethanol and biohydrogen, and also hydrothermal processes, that mean hydrothermal carbonization or liquefaction. So all these valorization routes have been reviewed recently in a paper entitled New Opportunities for Agriculture Digestate Valorization, Current Situation and Perspective. So in our laboratory, we are investigating the combination of the anaerobic digestion process with a thermochemical process called pyrolysis. So which is interesting is that during the anaerobic digestion, you produce electricity, but also you produce heat through the cogeneration heat and power system. If the electricity is sold to the public grid, the heat is generally loose due to the geolocalization of farm scale. So one idea is to use this surplus of heat, which is produced, to dry solid digestate to perform after a thermochemical process to produce some energy carriers, syngas, bioil, but also to produce biochar. So here is the result of the energetic and mass balances of the dual anaerobic digestion pyrolysis process on 1 day running. So as I said you previously, the methane produced, so this was the methane produced from a full-scale anaerobic digester. The methane produced is converted through a CHP system in electricity, but also in heat. We have shown that the heat surplus which is produced, it's enough to convert a total drying of the anaerobic digestate, or the solid digestate which is produced. Thus, after we can perform a pyrolysis process. Through this pyrolysis process, we obtain an excess energy from gas valorization, but also from oil valorization. And we estimated that through a dual system anaerobic digestion pyrolysis concept, we can increase from 42% the daily electricity production compared to anaerobic digestion alone. But we also produce a residue called biochar. Biochar can be valorized now from several points of view. One point of view can be the use of biochar for salamanders, but there is also a huge interest today on using biochar as bioabsorbent. It's the next step of our study. We have investigated the possible use of this activated biochar which are produced. So we have the process of anaerobic digestion. From the dry solid anaerobic digestion, we produce the core biologically activated pyrochar. It has been shown in literature that to perform first a biological pretreatment like anaerobic digestion process permit to obtain further pyrochar with higher porosity and accessible surface area, which are very important parameters in the case of bioabsorbent. So after we have used this pyrochar for detoxification of lignocellulosic hydrolysate, so a lignocellulosic hydrolysate is mainly composed of soluble sugar, but also of furanic compounds, mainly furfural and hydroxyl methylfurfural, that has been shown to be inhibitory of bioethanol process. So we have used this biologically activated pyrochar for detoxification step and after fermentation ethanol. So here are the main characteristics of the biologically activated pyrochar. So we can see that we have an interesting accessible surface of 88 meters square, and the pyrochar was mainly composed of microporous structure. So it's also important to notice that this value is very, very lower than classical industrial activated biochar that can exhibit accessible surface around 1,000 meters square. So here are some microscopy analysis. You can see easily the different micropore which has created in the pyrochar during the pyrolysis process. So in the next step, we have first analyzed the impact of using this kind of pyrochar for the detoxification of lignocellulosic hydrolysate. We have first worked on a synthetic medium containing 10 grams of glucose, 1 gram of furfural, and 1 gram of hydroxyl methylfurfural. First, we look the pyrochar concentration on a detoxification test of 24 hours. We increase the pyrochar concentration from 5 to 60 grams per liter. And we can see that around 40 grams per liter, we have excellent removal of both furfural and 5-hydroxyl methylfurfural. It's also, if it's not represented, it's also important to notice that the sugar concentration remain intact during all the absorption process. After we look at the concentration of 40 grams per liter, the time absorption, and the result which is interesting is that furfural is absorbed onto the pyrochar at a faster rate than hydroxyl methylfurfural. This could be explained by the difference of solubility between hydroxyl methylfurfural and furfural in water. The solubility of hydroxyl methylfurfural is higher than furfural in water. So after we tested the detoxification process on a real cornstalk hydrolysate, which was produced at a temperature of 150 during 30 minutes and a concentration of acid sulfuric of 5%. So the main characteristic of this cornstalk hydrolysate are soluble sugar concentration around 9 grams per liter, and it's interesting to see that we have mainly furfural production due to hemicellulose degradation during the pretreatment, and we obtain a concentration around 3 grams per liter. So here are represented the performance removal efficiency on the real cornstalk hydrolysate. So we can see that even in a real cornstalk hydrolysate, this pyrochar was effective in removing both furfural and hydroxyl methylfurfural. And the soluble sugar concentration was removed from less 5% from the initial cornstalk hydrolysate. So it's a very interesting process for bioethanol detoxification. So in the last step, we try to investigate the simultaneous detoxification and fermentation So for this, we use a synthetic medium. We use a concentration of pyrochar of 60 grams per liter, and we use 2 ethanolic strains at a concentration of 30 grams per liter. The first one was the Saccharomyces cerevisiae red ethanol, and the second was Saccharomyces cerevisiae leivine. So we have made 2 tests. In red are represented the ethanol production where the pyrochar was added in simultaneous with the fermentation, and in gray is represented the ethanol production and the furfural removal where no pyrochar were added. So the first result, which is interesting, is to see that in the case of pyrochar addition, there is a significant increase on the production ethanol rate, but the final production ethanol was the same. This could be explained by the high ethanol strain that we use in this study, which was around 30 grams per liter, and it has been shown in literature that Saccharomyces cerevisiae at high concentration is able to convert finally the furfural into less inhibitory compound called furfuryl alcohol, and these results are still in analysis, but we estimated that in that case, the furfural is probably degraded into a less inhibitory compound called furfuryl alcohol that permit after the ethanol production. So we will repeat this test using an ethanol strength around 10 grams per liter, and in that case, maybe we will see the inhibition process. So in conclusion, we can say that the dual anaerobic digestion pyrolysis led to an electricity increase of 42% compared to the anaerobic digestion alone. The pyrochar from digestate were selective toward furan compounds, and furfural compound was absorbed at a faster rate than hydroxyl methyl furfural compounds. Finally, the simultaneous and fermentation process was efficient in improving the ethanol production rate, but not the final ethanol production. As perspective work, we plan to test again the simultaneous detoxification and fermentation process at lower ethanol strain concentration. We plan also to study the impact of mechanical activation by using ball milling or vibro ball milling on digestate pyrochar characteristics. The idea is to find some green activation that don't require the use of chemical, and we could expect by using ball milling or vibro ball milling to increase the accessible surface and porosity of the pyrochar. And finally, we want also to extend the use of such pyrochar to the absorption of phenolic compounds, which can also be generated during lignocellulosic pretreatment, and among them we can find vanillin and cycloaldehyde. So thank you for your attention. Welcome.
Okay, thanks. So our next speaker is Dr. Sam Bessit, and from INRA, France.
Okay, thank you, Mr. Chairman, and good morning to everybody. I'm Cecilia Sambusetti. I come from France, but I'm Italian. And I'm specialized in biological conversion of lignocellulosic biomasses to produce biofuels. And today I will talk about the application of thermochemical pretreatments for the combined recovery of extractives and bioethanol production from softwood bark. So when we talk about the combination production of ethanol and extraction of molecules, we refer to a biorefinery concept in which biomass is not, and lignocellulosic biomass in this case, is not a residue but a resource to produce the fuel, chemicals, materials, and the renewable energies through both a biological platform and thermomechanical platform, or combination of them. But lignocellulosic biomass is a very complex structure composed of cellulosic structures that are interconnected by a network of hemicellulose embedded by lignin. Lignocellulosic biomass is also composed in terms of chemical composition of three major components: cellulose, hemicellulose, and lignin, and other components in a smaller amount, such as pectins and proteins. And the chemical composition of biomass, of lignocellulose, is generally depends on the type of biomass, but also the variety. However, due to its complex structure, lignocellulosic biomass is its conversion into biofuels is limited by its structure. So normally a pretreatment step is often required in order to degrade the lignocellulosic complex structure, decrease the crystallinity of cellulose, solubilize lignin, increase the accessible surface area and porous volume of the biomass in order to render more accessible to enzymes and bacteria the lignocellulosic structure. In terms of pretreatments, they are generally divided in three categories: physical pretreatments, chemical and biological pretreatments, and each pretreatment category has different effect on the lignocellulosic structure. In the table we can see different effect, the main parameters that can affect the enzymatic conversion of biomass into biofuels. But there are also some drawbacks about pretreatments. In fact, the pretreatment step is usually a high cost step in the biorefinery chain, and also some pretreatments, such as, for example, pretreatments that necessitate of high temperature or high acidic or alkaline concentration, can produce some sugar degradation products or lignin molecules from lignin that can inhibit the fermentation, the ethanol fermentation. So if we consider here the usual process scheme to produce ethanol from softwood biomass, we can see that we can use the softwood, the barked or barked biomass to produce ethanol, but it's necessary a pretreatment step, usually a combination of mechanical and physical chemical pretreatment, followed by an enzymatic hydrolysis and fermentation that can be performed in simultaneous step or in separate hydrolysis fermentation step. The idea is that if you use is not economically available, use the barked wood, and bark is usually separated from wood but is burned. So the idea is to use the bark to produce ethanol in order to valorize the residue. In fact, bark constitute a significant amount of the total weight of a tree, around 12%, and so it's important also to valorize this type of residue. However, bark, compared to wood, is a very different composition. In particular, has a higher lignin extractive and ash content that can limit its conversion into biofuel. extractive are the natural extractive present in bark are usually fatty acids, oleoresin, waxes, and phenolic compounds that in nature protect the tree from microbic and insect attacks. So it's important during the process scheme to separate them from the biomass in order to produce bioethanol then from bark. So the objective of the work was the evaluation of the effect of different pretreatment, so organosolv, diluted acid, and the combination of them on chemical composition of softwood bark, and then evaluate the feasibility of ethanol production and the influence of the residual extractive on ethanol fermentation. So first, softwood bark was cutted milled with a cutting milling with a 2 mm screen size. Then it was pretreated by an organosolv diluted acid pretreatment. Then the mixture was separated into liquid and solid fraction, and the extractive from the liquid fraction were characterized, and the solid fraction was used to perform ethanol fermentation by simultaneous saccharification and fermentation. In the table are summarized the parameters used to perform pretreatment. In general, we perform two temperatures. We tested two temperatures, 150 and 180 degrees. The reaction was performed for 1 hour. And here you can find the dosage of acid, sulfuric acid and ethanol with a solid loading of 100 grams of solid matter per liter. Ethanol fermentation was performed in simultaneous saccharification and fermentation, so by adding enzyme with yeast. We used an yeast Saccharomyces cerevisiae, which is able to convert on lexose and was produced by our team. We performed tests at 40 degrees for 72 hours at a pH of 5 but under stirring condition. And monomeric sugars and ethanol were analyzed by HPLC. So in this table is summarized the chemical composition of bark. As we can see, bark has a high lignin content that can limit the fermentation, and also a high dichloromethane extractives. These are the extractives analyzed by GCMS. As we can see, the majority of extractives are composed of polyols, but also a high percentage of fatty acids and ready acid are inside. And this is the composition of the solid pretreated separated residues after pretreatment. As we can see, as expected after organosolv pretreatment and diluted acid organosolv pretreatment, we had a solubilization of lignin, and only cellulose remained constant, while hemicellule was solubilized and degraded only after diluted acid pretreatment at high concentration. This decomposition of extractive recovered in the liquid fraction, and we see that after organosolv and diluted acid organosolv pretreatment, we join extraction of about 40% of extractives. So it's significant. About the results of fermentation, we can see that yeast quickly consumed free glucose after inoculation, and more than 90% of the ethanol was produced during the first 48 hours of fermentation. but in both in the case of untreated and pretreated samples, the ethanol production was very low. We join only 80% up to 80% of the theoretical conversion of glucose. This means that the limiting step is not the fermentation, but the hydrolysis, the enzymatic hydrolysis. So with this result, we can conclude that yeast are not inhibited by natural extractives, but the enzymatic hydrolysis is the limiting step, remains the limiting step. So for concluding, in terms of chemical composition, all pretreatments led to a solubilization of lignin, tannin, and suberin. Cellulose was not solubilized after pretreatment, and the amount of extractive originally present in the untreated bark were not totally solubilized, but we can organosolve and diluted acid organosolve pretreatment led to a high release of extractive, and according to fermentation results, in all cases, the yeast quickly consumed free glucose after inoculation. So these results suggest that no inhibition of fermentation occurred, but the enzymatic hydrolysis remains the limiting step for converting bark into ethanol. I would to thank Futurol project for supporting my research, and to BPI France for the financial support to the project. Thank you.
Nice presentation. Any questions? Okay.
So for the extractives, how do you separate them? How do you get the different components of the extractives?
After pretreatment, I separated the liquid and solid fraction, and then I analyzed with the GCMS the liquid fraction to characterize the extractives.
More questions? If not, let's thank Professor Sempesh again. Our next speaker is Professor Godiva again. I believe it's Professor Sanchez, and from University of Los Angeles in Colombia.
Oh, well, good morning, everyone. First, let me say thank you to the organizing committee for the invitation and to give me this opportunity to present some of my recent results. The title of my talk is Atom Economic Synthesis of Amides. And you already know how important amides are. We have two excellent lectures, one yesterday and the other one on Monday. So I don't need to remember you why it's important to find new synthetic methods for amide synthesis. They are present in a lot of natural products and pharmaceuticals and cosmetic industry, material industry, and among other fields. So to be honest, all this data about amide, we didn't know about that before we started this project. We started making something with glycerol. Actually, the idea was to try to make something new with glycerol. As you know, glycerol is a secondary product in the biodiesel production, so the production of glycerol is increasing year by year, and we strongly believe we need to make something different besides its use in cosmetical and food industry. So I am synthetic organic chemist, so I was looking for something to do with glycerol in synthesis. And I found a very old reaction, which is the scrub synthesis of quinolines. And many people around the world are working in this reaction, but using alpha-beta unsaturated aldehydes or ketones instead of glycerol. The reason is the dehydration reaction between glycerol and sulfuric acid to produce acrolein is violent, so you need to work with something safer. You also need, when you work with glycerol, you need some additives, and people use boric acid and also some carboxylic acids, and they are supposed to be a source of esters in order to make this dehydration reaction safer. And there is also the use of iron 2 sulfate, and it is supposed to be an oxygen trap. Nobody says how it works, but everything is an oxygen trap. You have a problem here. The oxidizing agent to generate the double bonds in the quinoline ring is nitrobenzene, and this nitrobenzene, once it's reduced, you have more aniline, and this aniline can also be transformed to quinoline. So you don't have a proper manner to calculate your yield in this reaction. And the reaction usually have very low yields. So we have some results about this, but I'm not going to present that today. Just one unexpected results we have. When we were trying to change the oxidizing agent, so we use iron 3 sulfate, and after 1 hour you have 89% yield of this formulated aniline. So too many questions were in our mind in that moment. Two of them was, is this a simple formulation reaction, and is the formyl group coming from the DMF? So we designed 2 different experiments, avoiding all sources of carbon besides the DMF. And you can see when you use boric acid after 72 hours, you have your product by TLC, but you cannot, you have less than 10% conversion. But in 8 hours, you have 60% yield of your formulated aniline. So the answer of these questions are yes, the formyl group is coming from the DMF, but this is not a simple formulation reaction, this is a transamidation reaction. So we decided to study a little bit more the scope of this reaction, and you can see 3 important things here. First, you can round your reaction in any kind of solvent. You can use polar solvents, less polar solvents, even protic solvents, but the reaction works better when you use non-polar solvents. And another thing important is when you use completely dry conditions, you dry your solvent or you use anhydrous salts of iron, the reaction time increases and the yield drops. So that means water is essential for the course of this reaction. We used different salts of iron, and we found nitrate was the best one for the reaction. To see the scope of this method, you can see here, you can use aromatic amides, aliphatic amides, secondary aliphatic amides, and even tertiary amides. And also it is working for urea and phthalimide, which is a good method for protection of primary amines. So, as you can see here, the reaction can be performed using a stoichiometric amount of both reagents, but the reaction time is a little bit longer and you have a lower yield. You also can change the proportion of the amine and the amide. Actually, I know amines are usually cheaper and more easily accessible than amides, but for local legislation in my country, for us it's more easy to find amides than amides. So that's the reason why we always use an excess of the amide and not the amine. Then continue with the scope of this reaction. You can use it as a very good method for formulation. You can use aromatic amines, substitute aromatic amines, heteroaromatic amines, or even more complex heterocyclic amines. You can use secondary amines, aliphatic, and even bulkier, say, aliphatic amines. This is a bond missing here. So when you also have two nucleophilic groups in your substrate, the reaction is, as expected, selective to the nitrogen, the more nucleophilic nitrogen. This is one of the few examples for transamidation reaction who works better or who works well with all the amino acids. The only thing you need is the ester of the amino acid. You can also use, as I said, more bigger aliphatic amides. In cases of aromatic amides, you have not always good yields. In some cases you have a very good yield, but in some cases you have very low yields. As I said, you can use it as a protection reaction for primary amines. And with ureas you have, when you use primary aliphatic or aromatic amines, the reaction works very well for the desubstituted urea. But when you use secondary amines, the reaction stops even with an excess of the amine, stops in the monosubstituted urea. So we use this reaction to make mixed or asymmetric ureas. You can start with the primary amine or with the secondary one. You just look your TLC, and when you have complete conversion of the amine, you add the second amine and 5% more of catalyst, and then you can have your product in a very good yield. We propose a reaction mechanism, and actually there is not the only transamidation reaction reported in literature. There are some other examples, but always when people work with metals, they have basic ligands. So the only difference with this mechanism and the mechanism reported by Professor Stahl is we propose the role of water is to take the proton of the amide to start the reaction cycle. Here, A complex is supposed to be more stable when you use aromatic amines than when you use aliphatic amines. That's the reason why when you have aromatic, it's more complicated to perform this reaction. When we try thiourea or thioacetamide with any source of iron, the reaction was unsuccessful. So we also, in our optimization experiments, we use other sources of other metals. And we were happy to see that antimony 3 is a good catalyst for this thioacetylation reaction. Unfortunately, as I just say, antimony is not a good example for green chemistry, but it's working very well with with aliphatic primary amines and also with secondary amines. When you use aromatic amines, the reaction somehow has problems, and you need to add your amine slowly. So we use a syringe pump and we add the amine in 10 hours, and then you just let 2 hours more to the reaction finish, and you can isolate your acetylated aromatic amine in a good yield, or acceptable yields. Then we wanted to apply these transamidation methods for the total synthesis of some natural products, but we realized that it is not usually easy to find the amide. So it is more accessible, the carboxylic acid. So we were looking for one method to make amides directly from amines and carboxylic acids. You can make a parallel with the esterification reaction, and you only need to take off water. So there is one study of Professor Lupi in 2002 where he hit amine and carboxylic acids under microwaves, but the reaction was limited to the use of aliphatic acids and amines. Other two examples used some mesoporous catalyst, one of them prepared from silica gel. So we were happy to see that when you use silica gel as a solid support and you heat the mixture under microwave irradiation 2 cycles of 20 minutes each one, you have a very good yield of your amide, and you don't have traces of the Michael addition product. So that's the reason why we use cinnamic acid as a model for this reaction. You can see the scope here. You can use any acid, small carboxylic acids, bigger carboxylic acids, even very acidic carboxylic acid as trifluoroacetic acid or benzoic acids. You don't have problems with double bonds. You can have also fatty acids, and it works very well. You can mix acids or amines, whatever you want, and you have very good yields. The only problem you have is when you use bulkier secondary amines. In that case, you only can perform your reaction with small aliphatic acids and in a very low yield. This is a successful example using a secondary bulky amine. You can, as I say, use also unsaturated, beta-gamma unsaturated acids, and you don't have the conjugation product as a major product. You see that in by NMR, but it's only 15% or something like that. So you can still isolate your beta-gamma unsaturated acid as the major product. We make some experiments to see the selectivity of this reaction. You can add a nucleophile in the acid. Of course, we help a little bit the reaction, adding a little excess of amine, and you have only this product. You don't have the homocoupling, not the ester here, not the amide here, but not the homocoupling product. When you have a secondary nucleophile in the nucleophile in the amine, you can also see the reaction is selective to the nitrogen. In this case, we are not very sure. You have the dehydration product, and we don't know if the reaction runs for the amidation or the deesterification first. The reaction is selective to primary aliphatic amines. You can see that in these two experiments, when you use aliphatic and primary and secondary amines, you only can isolate the product from the primary amine, and when you mix aliphatic and aromatic, you only have also the product from the aliphatic amines. So in summary, I showed you 3 different methodologies to make amides, one using iron 3 and water, simple salts of iron 3. The second one to make thioamides using antimony 3 as catalyst, and the third one using microwaves and silica gel as a solid support. So let me say thank you to my co-workers. These are former members, these are current members who actually are in the picture here. People in blue was working directly in this work. My work is financed by two government institutions. This one first for the fellowships of students, and the enterprise extra for the kindly donation of some chemicals. Before finish, I just want to show you some pictures from the university. This is a very young university, around 50 years old. We have a beautiful campus here. We are located at Bogota. Bogota is in the middle of Colombia. Colombia is in the north part of South America, and we are in the middle of the mountains, so we are 2,600 meters above sea level. So some people say it's very cold in Bogota, but we used to say we are closer to the stars. Yes. Thank you for your attention.
Thank you for a very nice talk, and also I'm very impressive with your university.
Thank you.
And we can open the floor for questions.
In the transamidation, in one case, you use dry toluene.
Yeah.
And you said that the yield dropped.
Yeah.
But you have water in the iron salt.
No. In that
Yes,
case,
this is a hydrated
yes.
iron nitrate.
But for this for that example, we use anhydrous iron chloride. So
So I didn't.
yeah.
I didn't.
So
catch.
yeah.
Okay, thank you.
Okay, yeah, I want to ask about something. It is well known that amide function group should exist in equilibrium with its tautomer.
Yeah.
So, how you study this, the effect of this phenomena in your interactions or something?
No, we didn't know.
So
we
in
don't know.
particular, you worked in mixed solvents, so in protic and non-protic. So this is a phenomena could make a significance for you.
Maybe, yeah, but we don't know, study the mechanism
Okay.
yet. But it's something we have to do.
More questions?
In your microwave method, what was the temperature? Because
130 degrees. So you have to heat 130 degrees. You make 4 cycles of 20 minutes each one. You have just to heat 20 minutes. You take your flask off. You dry a little bit the water and you put it again, and it works.
so is it okay? Because I'm confused, because then you take it off to remove the water, and then
But you just remove the water manually. You can take a small piece of paper and take the water and put it again. It's the same temperature when people heat these both reagents without the silica, and you have an increasement of the scope of the reaction and also in the yields.
what kind of microwave did you use?
It's a SEM microwave reactor.
Okay.
You can also do it in a home.
Well, with the homemades, you cannot control the temperature.
Yeah,
that's
that's a problem.
Especially you have
when
a
you use-
silica gel.
Yeah, when
Then
you
you
use
should-
a home made microwave, the reaction is finishing after 20 minutes. But of course, you cannot reproduce the results very well because you cannot control the temperature.
Yeah, and in the mechanism that you showed for the first reaction,
Yeah,
you said the water removed proton from the amide.
that's the we propose this.
What's the pKa of the amide?
The pK of the amide should be 17 or something like that. It is of course an equilibrium, but the equilibrium is displaced because of the reaction itself. So as you can have more steps here, so that's why the reaction is that is the force to drive the reaction. And you need, of course, an excess of one of the reagents to push the reaction.
Okay, thank you.
Comment about the pKa of amide. The pKa of amide is so high, so it is not usual to ionize in neutral or in water. He cannot give proton to water. He can get proton from water.
Yeah.
Yes, but you know, this is not completely water. There are some organic solvents and non-polar solvents, so the equilibrium and the dissociation is different.
All right, you guys can continue the talk after the meeting. But that'll bring us, thank you for Professor Sanchez and for your nice talk. And that'll bring us to the last speaker for this morning. Who? Oh, she's not here?
So she's ready.
She's ready, but maybe I can ask, you already talked to her?
Yeah.
Or do you want to bring this person up? Because
No, I already spoke to her.
Oh, you already spoke to her, okay.
because of people has to travel and so on, Hui Song is going to be the first. She's from the Chinese Academy of Science in China, from this Tianjin Institute of Industrial Biotechnology, and she's going to speak about industrial enzymes. So please.
Thank you very much for the introduction. Also, thank you for the community give the chance for do the presentation today. My work mainly focus on the industrial enzyme discovery and engineering, but the purpose of this is finally to do the application. So today I'm going to talk about the industrial enzyme used to build green technology for pretreatment of textile dyeing. First of all, I would like to give some brief introduction why we do this in China. Oh, sorry.
This is the pointer, but you should press very strongly.
Okay.
So this is to go back and forth. Okay, let's. Okay. Come on. It's okay. Let's go back. No, this is not yours. I don't know why it disappeared. No? What happened?
Okay. If this is a PDF, you can just touch screen it. You're gonna have to do that. Okay. To your next slide.
So this is not mine.
It's not. No, it's not.
Okay.
This is not the right presentation?
No.
It's not mine.
Okay.
Sorry. So first, I will give the brief introduction of the background. In China, the textile industry is a pillar industry, but also is urgent demanded for energy saving and reduce the emission of the industrial. For the discharge water in textile industry is about 1.1 billion tons. It's about 6.5% in all kinds of the industrial wastewater emission. But also the COD emission is 30 million tons. It's about 5% of total industrial emission. So this industrial really needs some green technology to replace the traditional process. So the printing and the dyeing process is one important part for the textile industry, including three sections: pre-treatment dyeing and printing, dyeing, and finishing, in which the largest emission part is dyeing and printing process, pre-treatment process. Let's see the traditional process of before dyeing. Normally, also there are three processings. First is designing, and second is called scouring and bleaching. But in the traditional process, all of them we can see in a very high temperature at 90 degrees. Also, it uses very concentrated alkaline and some refining reagents. And also, in the bleaching process, they use hydrogen peroxide. So they use a lot of water to remove that. So this is a really high energy cost and large amount emission part. So the industrial enzyme can use to replace this process to set up a green craft. In fact, the designing processing is used to remove some slurries. It's containing some starch and PVA. And the screwing process is used to remove some impurities like pectins, horny, waxy, hemicellulars. So these materials can be directly degraded by the enzymes. Also, the splitting process can be used to catalyze to decompose the hydrogen peroxide into water and hydrogen. So the industrial enzyme is really useful tool to set up this green technology. But still, we need to face and solve some problem when the industrial enzyme to apply for this process. The first, the enzyme working always has restriction for at pH and temperature. So how to make the enzyme can working in same condition, in certain conditions with high activity? This is we need solve the first issue. Second one, the enzyme using always more expensive than the chemicals. So how to reduce the enzyme using cost? This is second issue. The third one, in the whole process, one single enzyme cannot solve the problem. So we need complex enzyme together to make all the enzyme working in the same conditions. To solve this problem, a lot of technique can be used with the development of the biotechnology. So first we can engineer the enzymes and make molecular modification to change the property of the enzyme, make them working in the certain conditions. Also, we can use the rational design by the study of the structure of the enzyme and directly change the property of the enzyme. Finally, also we can use in bioinformatics coupling with computing design, make new functional enzyme. How to reduce the enzyme cost in application? So we must set up an overexpression system to overexpress large amount of enzyme. Also, we need to optimize the fermentation process for the enzyme and make the production cost very low. And finally, we need to set up the good method for the enzyme preparation and separation, make it stable. So how to make the enzyme working together? We need to complex enzyme, which should evaluate exactly but in the actual industrial application. So we can summarize the technology roadmap like that. First, we do molecular modification for enzyme, then high expression for that, reduce the cost, and to scale the preparation and for large amount enzyme preparation, finally for industrial application. So what we have done and what we have achieved. In our institute, we have a really good high throughput screening platform. So in this one, so the whole process can be automatic for sample collection, separation, and the performance testing and rescreening enzymes. So the capacity of the maximum number of weekly response can be 57,600. And annual operating recorder number is 30 million times. Colony pickup capacity is 10 million times. So screening with high expression engineering the bacteria can be 5 to 100. So this efficient high throughput platform can help us to obtain large amounts of enzymes which has a potential in the industrial application use. Also, we want to find some new genes of enzyme. And the best way, we can use the environmental samples. We collect some alkaline soil samples. Then by the metagenomic sequence technique, so we use bioinformatics study and to analyze the data. First, we know the bacterial distribution in this samples and make environmental genomic library and do the screening. Finally, we get near 1,000 of the new enzyme genes and also more than 150 enzymes can relate it to the textile application. We also construct some expression systems for enzyme expression. Finally, we find some important enzyme expression can be used E. coli. That is a really cheaper expression system. By the optimization of the fermentation process, the catalyze and the pectinase could be in the fermentation broth, the activity can reach to 80,000 units per milliliter and 106,000 units per milliliter. That will be really decrease the use of the enzyme. It's really good for the industrial application. Finally, we form 3 complex enzymes by the evaluated by the actual industrial production. So these 3 kinds of compounds, enzyme for pre-treatment of dyeing is for 3 steps. The complex for designing enzymes is mainly complex with the amylase and the PVA degrading enzymes, and their working temperature at 7 to 9. The pH is working at 7 to 9. Temperature is about 45 to 60 degrees. And also the screening process enzyme is some pectinase, hemicellulose, and the lipid something to complex together. The bleaching process, we use catalyze to breaking down the hydrogen peroxide. We use these 3 product enzymes to set up a green process for the textile pre-treatment of dyeing. From this slide, we can see the enzyme can completely replace the chemicals in the traditional process. Also, the temperature from the 90 degree decreased to the 50 degree. And we're using catalyze to breaking down the hydrogen peroxide. At least can save 50% water in use. Also, we can turn on the 2 steps, designing and screwing, into the 1 step. So this saves a lot of the production period. The enzyme green, the enzymatic green process is not. We can use a lot of textiles pre-treatment process. We can successfully use to polyester Kevlar heat cable, because this is textile, the name. Sometimes I don't know how to pronounce it. And also some pure cotton clothes and polyester and cotton banded fabric. So we can use this process to treat all of them. So this, sorry, this is. We also compare 2 processes. What's the cost? This table list is about 10,000 milliliter of the fabric treatment, the cost in the traditional process and enzymatic treatment process. From this table, we can see the cost, the enzyme process, enzymatic process can be reduced from 4,000 to 2,000 RMB. So it's at least 50% saving in this enzymatic process. Also, we compare the quality of these 2 processes and the COD emission amount. From this table, it's very clearly the COD emission really has a great reduced. So we can summarize these two tables into several points. So the enzymatic process saves 60% in production cost and 35% in energy water consumptions. The second, the products of enzymatic process improve stretch strength 5% in longitude direction, 15% in latitude direction. Break off strength 10% in longitude direction, 11% in latitude direction. After enzymatic process treatment, dyeing is more pure and shady and have high delta E value. So the COD volume decreased 85% in enzymatic process. So the enzymatic process is not only can use to the textile industrial, also it can use in the paper leather industrial as well as the gelatin manufacture and other traditional process. So the industrial enzyme have widely range of application in establishment of green technology. Thank you very much for your attention. I have to shorten my presentation.
No, thank you. Okay, thank you very much for this kind lecture, this nice lecture about the enzymes application. Some question?
So can you tell me, this is very nice work here, so can you tell me about the working condition of your enzymes? Usually, what is the temperature range and what kind of solvent?
So normally the working condition for the enzyme, in the library, they use the room temperature. Sometimes it's 37 degrees, it's optimized temperature. But for the industrial application, always in a very high temperature. But at the beginning, we select some very high temperature. The enzyme has very thermostability, can working at 90 degrees. But we think if we use these enzymes, it's no useful to save the energy. So we select some enzyme to do the direct evolution or even the rotational design to make the enzyme working condition in the 45 to 60 degree. That I think is acceptable for this condition for industrial. And
more questions? If this is not the case, let's thank again the speaker for the nice talk.
Thank you very much.
Hello everyone, my name is Ewa Bogel-Łukasik. I'm working at New University of Lisbon. that is Associated Laboratory for Green Chemistry in Portugal. My new laboratory is called Advanced Fluids in Tailor-Made Biofuels and Bio-Based Product Processing. This work is focused on alternative ionic liquids-based lignocellulosic biomass pretreatment and fractionation towards progress in biorefinery. And it's a joint work with other Portuguese centers and Brazilian centers, and was financed by different Portuguese and European projects. Considering the European technology platform supply chain for 2015 and beyond, we can see that we have a long way from farmer to food manufacturer to food retailer, and then for us to consumers. Why biomass is so important? Because it's widely available, and it's cheap, and it's a good source of energy for all kinds of activities. It regulates essential activities of human systems, and plants are full of many fundamental chemicals. Who is interested in biomass raw materials? There is many sectors who wants biomass. Biomass finds application in food, feed, biochemical, and energy application. At the best guaranteed price, it means for free. And there is a big competition between food and feed production and also biofuel production. It means in transport sector regarding use of agricultural crops. For that moment, biorefinery is using biomass not very broadly, and now it's indicated that is the area marked in dark blue, but the future of biorefinery is envisaged to be much broader, and it's indicated in light blue color, light green color. As we know, lignocellulosic biomass does not have a simple structure. It's a complex biopolymer, and it's composed of cellulose, hemicellulose, and lignin at different percentage depending on the feedstock. Cellulose, it's mainly homopolymer of glucose, and it's very difficult to hydrolyze. Hemicellulose, it's a heteropolymer mainly of pentones and hexose, and it's easier to make the polymerization. Lignin has aromatic structure of cross-linked polymers of phenolic monomers, and it has completely different depolymerization chemistry. On these slides, it's illustration of micro and microfibrils of cellulose, and they position in the walls just to see the complexity of the cellulose. And cellulose polymer chain also, they are able to create hydrogen bonding between the chain in the heteromatrix of the cellulose. So pretreatment of lignocellulosic biomass is essential to disrupt the complex structure of biomass, to allow to extract lignin and decrease cellulose crystallinity, to increase surface area, to allow for hydrolysis. Up to that moment, they are pretreatments of biomass, which are divided in 4 groups, like physical, chemical, physicochemical, and biological. And here on the right-hand side of the slide, we can see the structure of biomass without the pretreatment and after the pretreatment, showing the disruption of the structure. Biorefinery is a facility that integrates biomass conversion processes and equipment to produce from biomass chemicals, energy, fuels, materials, And green chemistry that is based on 12 principles has a great interest, and it's good to combine these two fields in order to develop more sustainable processes of the future. Among common methods of biomass pretreatment, recently ionic liquids found very increasing interest in this field. According to Easy Web of Knowledge, the topic of ionic liquid is visible more than 100,000 times, and the topic ionic liquid and biomass is shown more than 1,500 times in a period of 9 years. And topic of biomass and ionic liquid already was cited more than 31,000 times with an impressive H index of 80. So it means that one of the possible solutions in biomass pretreatment might be ionic liquids. Interest of ionic liquids used in biomass mainly depends on their solubility, on possibility to make efficient pretreatment and conversion of carbohydrates into value-added products. Solubility and pretreatment of biomass with ionic liquids depends on the ability of an anion and cation of ionic liquid to interact with carbohydrates, water content of ionic liquid, and also mutual interactions in raw biomass. Ionic liquids are composed of cations and anions, and here is illustrated only some of them most commonly used. And as we know, there is numerous possibilities to synthesize ionic liquids, like we need that we need for a specific application. Ionic liquid possess exceptional properties such as high polarity, negligible vapor pressure that eliminates risk of explosion, thermal stability, conductivity, and the most important is solvent power and tunable solvation that can be obtained by the synthesis of a task-specific ionic liquid. A lot of properties of ionic liquid can be tailored, and we can produce ionic liquid that we need for a specific application. Ionic liquid used in the pretreatment of biomass, they have possibility to alter the physicochemical properties of macromolecular components of biomass, extract a specific fraction, and perform fractionation after dissolution of biomass. Its advantages because we can obtain decrease in crystallinity of cellulose, increase extraction of lignin, lower degradation of monosaccharides, and sustainable aspect also due to ionic liquid reuse and recycle. We have performed pretreatment of biomass with ionic liquids. There were 4 different biomass: wheat straw, sugarcane bagasse, rice straw, and triticale straw. And ionic liquid that we use according to the solubility, it was amine acetate, bemine dicyamide, thiosanate, and hydrogen sulfate, as illustrated here. First biomass was milled to be prepared for pretreatment, and then here is illustrated the methodology A, that we made pretreatment of wheat straw with amine acetate at 100 degrees at 6 hours, with the ratio of biomass and ionic liquid 5%, and we used a the fractionation agent alkaline solution, and we are able to fractionate carbohydrate-rich material and lignin. Ionic liquid recovery at this stage, it was 73%. Then when we used acetone solution and decreased a little bit time, temperature and ratio of biomass and ionic liquid, we were able to obtain carbohydrate-rich materials and then to make fractionation with alkaline solution of cellulose to obtain cellulose, chemicellulose and residual lignin. The ionic liquid recovery was 93%. Then we came back to the method, to the advantages of method A, and this obtained from method B, and we use alkaline solution not only for the first part, but also we used after the regeneration, we used for fractionation of carbohydrate-rich fraction to obtain cellulose and chemicellulose and residual lignin. We made identification of all samples, carbohydrate-rich samples, also lignin. We made infrared spectroscopy, that we can see that lignin aromatic vibration decreases its intensity from the wheat straw, which is green, to other regenerated materials that we don't have. Also, ester linkage we have in wheat straw, and then we do not have in our samples. Lignin samples spectra look completely different. We also perform this analysis. So we got to the conclusions that method C gave us more pure samples of cellulose, chemicellulose, lignin, and we have lower loss of carbohydrates. So we used method C for wheat straw, for pretreatment of wheat straw with different ionic liquids. As we can see, we were able to regenerate cellulose and chemicellulose-rich samples. They are rich with carbohydrates, which are indicated in green, and lignin samples at high purity, carbohydrate-free. Then here is example of the sugarcane bagasse that was pretreated with amine acetate, and also after the regeneration we made the additional fractionation, and we were able to obtain cellulose and chemicellulose rich fractions rich in carbohydrates and also residual lignin. Here presented results for other types of biomass, and also it confirms the results that it was very important to perform fractionation of the liquid stream after the regeneration process in the pretreatment of biomass with ionic liquid. So we are able to produce cellulose and chemicellulose fractions at very high purity of carbohydrates and lignin samples without carbohydrates. To see the efficiency of the pretreatment, we perform enzymatic hydrolysis. And here are results for wheat straw, that we have wheat straw, then going to the right, wheat straw after acidic hydrolysis, then regenerated material from method E, cellulose-rich material obtained after method B, and then cellulose-rich material obtained after method C, that was this one, optimized, and standard of cellulose. As we can see in green for cellulose C, this indicates that we obtain highly digestible material. Also, we perform hydrolysis of the biomass, which was pretreated with BMEM hydrogen sulfide. And it was possible to notice that chemicellulose that was selectively dissolved in this ionic liquid, then when it was hydrolyzed, produced furfural and hydroxymethyl fural. Here are results of other enzymatic hydrolysis of sugarcane bagasse. Also cellulose CA obtained by pretreatment with ionic liquid indicates that we have highly digestible cellulose. The same we obtain in case of rice straw. It's cellulose CB. And triticale straw also confirmed these results, that the pretreatment made with ionic liquid was efficient. We calculated also crystallinity indexes, and first, as you can see, there are examples of lateral order indexes and total crystallinity index calculated for pure biomass, then biomass with AH, it means after acidic hydrolysis, then regenerated material of each biomass, cellulose standard, and cellulose rich material obtained after this optimized method C. And we observe that we have increasing crystallinity index. The trend of crystallinity index is decreasing, and we obtain the lowest values of crystallinity indexes LOE for regenerated material and cellulose rich fraction, which is indicated in yellow. So this confirms that our material changed the surface, and it was better accessibility towards hydrolysis that confirms the results of the hydrolysis performed. Regarding the recyclability of ionic liquids in the pretreatment of biomass, here I present the results of pretreatment with amine acetate, pretreatment of wheat straw. As we can see, we were able to perform 7 pretreatments with high ionic liquid recovery, that is indicated in pink color, and the yield of regenerated material was mainly the same. We did not lose any efficiency of the pretreatment. Also, we checked other ionic liquids, and we even were able to obtain ionic liquid recovery up to 97% with using of BMM dicyanamide, with also high yield of regeneration, more than 77%. So concluding pretreatment of biomass with amine acetate for all 4 types of biomass was efficient, and we were able to obtain cellulose, chemicellulose, and lignin at high purities. BMM hydrogen sulfate used in a pretreatment allow us to selectively dissolve hemicellulose and then produce value-added products after the hydrolysis process. We can conclude that also it's very important to make the fractionation of this liquid stream after the regeneration process, because we can recover the remaining amount of lignin and chemicellulose. We obtain highly digestible material in a pretreatment all biomasses with ionic liquids. We have the decreasing trend of cellulose crystallinity that made the cellulose surface of more accessible towards hydrolysis, and also hydrolysis results confirm this efficiency of the pretreatment made with ionic liquids. And also we were able to make recyclability of ionic liquids up to 7 times without losses in the efficiency of pretreatment of biomass with ionic liquid. And for wheat straw, it was 95% using amine acetate, and for example, for other biomass, it was even 97% when BMM disenemate was used. This work was supported by Portuguese Foundation of Science and Technology and many projects, also by cost actions, emergence of complex systems, and valorization of lignocellulosic biomass side streams for sustainable production of chemicals, materials, and fuels. And here is list the researchers and collaborators who were involved in this work. Thank you very much.
Thank you, Eva, for the nice talk. It's time for questions, comments.
Okay, thank you for your interesting talk. And actually, also I thank you for the real actions that we see, that how to deal with scientists and to deal with child at the same time. So it is fine, and control your feeling. Okay, I wanted to ask you about ionic liquids. You use the ionic liquids pure or mixed with water in your biomass production? Pure, I mean without water or in presence of water?
Because my background is physical chemistry.
Yeah.
So I learned from my PhD that first we need to release water, I made part of my PhD in Belfast and Queen's University of Ionic Liquids with Professor Ken Seddon. So first we need to put ionic liquids into high pressure to evaporate the water, yes. And we perform Karp-Fischer pretreatment titration to see the water content.
You know what is the reason? You know what is the reason for this? It's important.
Yeah.
You know what is the reason you need to evaporate water? You know?
Yes, yes.
You know. Okay, because you know the mechanism of ionic liquid in presence of water is completely different in absence of water.
Yes.
So it is interesting, and also the efficiency that you present is very high.
Yes, because we released water.
Yes, I thank you for your
We have, for example, like 150 ppm, 200 ppm.
Yeah, good.
Yeah.
Okay, thank you.
Another question?
Thank you. I have a question about your enzymatic hydrolysis efficiency. You calculate your hydrolysis efficiencies as weight by weight of biomass, your glucose yield?
Yes.
Yes, but you calculate also, you compare your glucose yield in terms of gram of glucose release per gram of glucose present in your biomass. You make a calculation about the glucose, experimental glucose obtained compared to the theoretical glucose present in your biomass.
I suppose so, but I'm not
because
sure.
It cannot be 100%. You understand the question? This is the glucose yield in terms of gram of glucose per gram of biomass. But my question is if you calculated also the experimental glucose obtained by hydrolysis compared to the theoretical glucose initial in your biomass.
I have to check this.
Yes, because it cannot be 100.
Yeah.
Okay, thank you. More questions? If not the case, thank you, Eva, for the talk.
Thank you.
Previous, next. Okay. Yeah. Very good afternoon, one and all. I am Dr. Praveen from Sahasra Pharmacy College, working as professor and principal. I'll be talking on green chemistry as a tool to prevent pharmaceutical hazards and pollution. Actually, the strong reason why I had chosen this topic was when I have taken my M.Pharm students to pharmaceutical industry for their project work. I had experienced a few cases in the pharma industry, especially in the research and development division. One of the employee, he was working for the past 10 years and he complained about loss of hair because he consistently worked on anti-cancer products. So after a thorough analysis was made, it was concluded that he had repeated exposure. So probably because there were proper measures not taken or the setup was not proper. And another person complained of skin problems, again the same reason. So now in pharma industry, green chemistry is emerging as one of the important concepts, probably apart from the drug discovery pipeline. So what I felt is, being in academics, and wanting to collaborate with pharma industries, I think green chemistry could be one of the potential topic in the future, and new changes can be done in this area in the pharma industry. So there is if you see the statistics, the amount of solvent usage, either it may be in the synthesis of various compounds or to solubilize various compounds, what kind of favorism the solvents are doing to the pharma industry. So right from the inception, that is 1975, to update, if we see relatively the amount of favorism and unfavorism, it is gradually decreasing. So you can see that the favorism is increased, again there is a gradual decrease, and again unfavorism is also falling. So this indicates that yes, this time has come up to reduce the amount of solvents that is being used, probably in the chemical industry or pharma industry. This is a clear evidence that is shown. Why the solvent usage is more? Because this is again a statistics showing worldwide that the amount of chemicals output in the industry is increasing day by day, because the demand is increasing for, and the chemical drugs or chemical substances which are being synthesized, some of them are being used in food industry, some of them are being used in plastic industry, some of them are being used in pharma industry. Therefore, the need for synthesizing chemicals in the chemical industry is increasing day by day, and for this synthesis the amount of solvent usage is increasing, and that's how probably various problems do arise. And yes, again the concept of green chemistry has to be put up. So this is again even in the highly developed countries like Japan and Europe and America, the chemical industry output is very high with regard to the usage of chemicals. So this is a picture showing on the growing incidence of environmental accidents. The moment chemical industry output is more with regard to the usage of solvents, probably indirectly it is an indication that the environmental safety is going to be a major concern, because you need to provide a good environment to the employees, as well as the working environment should be safe, so that it will be safer to the person working as well as the industrial setup. So here we can see 10% industrial pollution is contributing to 17%, traffic accidents is contributing to 18%, and production safety incidents with regard to chemicals is contributing more, that is, it is almost coming to 42%. Therefore, the production of chemicals which is leading to environmental accidents is very high in this statistical picture. So again, this is an indication that the environment is becoming unsafe, where the usage of solvents is very high. So some companies have already implemented the concept of green chemistry to cut down the environmental disasters, and we can see here the statistics among all the industries. The pharmaceutical companies, the wastage produced is 2.5 into 10 to the power of 5, because I am related to pharma company. I am just trying out to figure out what is the extent of damage that is occurring in the pharma industry in parallel to the drug discovery pipeline. So here the E factor is between 25 to 100, which is nothing but it is the total mass of waste produced, during the process of synthesis of a compound. So it is the responsibility of every pharma industry to have their own measures and calculations to bring down the E factor, so that the solvent toxicity can be reduced. So if you can see this figure, the amount of solvents used for active pharmaceutical ingredients, that is new compounds, is corresponding to 49%. It's very high. Like we can see dichloromethane, methanol, chloroform. And now all the pharmacopoeias are also releasing new literature in relation to the hazards that are being related to these chemicals and what are the different measures and environmental setups to be taken by the pharma industry. So now, based on these statistics, I think green chemistry can be considered as a very key role in the pharma industry. So green chemistry, as you all know well, it is nothing but the design of chemical products and processes that reduce or eliminate the use of generation of hazardous substances. So the application is that can be made at the level of education, educating the students, and this concept can be very well applied in the projects when given to the students, and the same thing can be applied at the industry level. So probably I think green chemistry concept can be collaborated between academia and pharma industry so that a proper balance can be made for the upcoming generation in the discovery level. So green chemistry can be considered as a pharmaceutical hazard and pollution free concept when implemented in the synthesis of compounds or solubilization of various substances. So green chemistry being the science, it can introduce a new substances into the world and we have a responsibility for their impact in the world. So a positive impact should be generated wherever solvents are to be used. So the control of solvents should be made so that there should be a limit in the percentage of solvents to be used. And yes, regulatory measures have to be taken and some mandatory measures have to be brought up so that probably every company or college can follow very strictly, apart from just putting on the paper. Cleanup. So cleaning is again an important factor. See, many of them, even I observed many students, after performing experiments, they just leave the glassware like that. So if proper cleaning is not done, again, in course of time, that can lead to generation of very toxic substances. So all these put together, you can inculcate the concept of green chemistry, ultimately giving a positive impact. So with these factors, probably 25% of the cost cutting can be made indirectly by any firm. So you are not investing, indirectly you are reducing the cost. So the fundamentals of green chemistry, it increases the awareness and understanding of green chemistry principles, alternatives, practices and benefits for all group of sectors wherever the solvent usage is being mandatory. And basically it can integrate the principles of green chemistry as well as green engineering. So now many new pharma companies are coming up. Probably the design of the industry can be made by discussing the concepts of green chemistry with the engineers. And then what are the different equipments that have to be used for the synthesis of chemicals. So all put together, the risk factor can be analyzed, and probably by a thorough analysis, it should be taken care that the percentage of risk level should be as minimum as possible within the given circumstances. There are lots of principles involved which can be applied in individual sector as and where necessary. Like you can prevent the wastage instead of calculating and reducing at the end. A proper analysis can be made in the beginning and the amount of waste can be prevented. And similarly, atom economy. There is one substance called artemisinin, which is one of an excellent anti-malarial. So it is an excellent Chinese anti-malarial drug, but now people started synthesizing this drug and it is taking more than 32 steps. So in this 32 steps, how many intermediate compounds are generated, how many of them are toxic, if they undergo degradation, what type of toxicity could be given, and what is the percentage of solvent that is to be used is going to be the biggest question mark, apart from obtaining a new molecule. So probably in atom economy, the number of steps could be reduced by redesigning, because we have got number of softwares which can be taken, where the help can be taken to minimize the number of steps. Then similarly, safer solvents and auxiliaries. Yes, you can select where some solvents where the toxicity level is very low and where minimum amount of solvent can be used. There are substances where the dielectric constant is more and you can use 10% of the solvent to solubilize almost 100% of the drug. So such combination can be selected to reduce the input of the solvent. And similarly, you have design for degradation as well as real-time analysis. Now, if you take HPLC, I think HPLC is one of the important tool for qualitative as well as quantitative analysis. So we require umpteen number of solvents for using HPLC also. So for that, again, redesign can be made in developing a safer mobile phase composition, so that water composition can be made, increased by reducing the solvent. So it all depends on a proper balance. It's not mandatory that every time you should reduce, but wherever is required, a separate analysis can be made by appropriate design. So if we look into the literature, the concept of green chemistry is increasing, and the evidence is right from the beginning. Even if you take in US, Europe, Japan, the most highly sophisticated countries, the amount of patents that are being filed year to year are gradually increasing. So that itself again an evidence that yes, green chemistry improvement can be done in the pharma sector especially. And if you look into the academia, even the number of publications are increasing enormously year to year, apart from pharmaceutical patents. Now, what are the key drivers or factors that is going to drive the role of green chemistry in the pharma sector? So the first one, as we have seen, that tremendous cost cutting can be done. So cost savings is almost up to 45% that can be made. Consumer awareness. Now every person is very much concerned about his health. So probably, for example, there is one product, doxorubicin liposome. So some companies claim that 0.1% trace amount of solvent could be present in the preparation. Now, if 0.1% of the solvent is present, and if he is going to use the product for 10 years, the amount of accumulation of solvent in the biological system is going to be a question mark. So probably that trace amount also should be removed. With regard to consumer awareness, so therefore the patient might not prefer in the future. Next is, as I have mentioned, regulatory aspects have to be increased further in official compendiums so that it will be mandatory followed. Development of new innovative products. So solvent cutting can be used even during the development of new products. Either a totally drug discovery can be made, or an alternative derivative can be synthesized, or any new substance can be directly extracted from a new plant. So especially if we look into extraction methodologies, aqueous based extractions are increasing day by day, and number of publications are being made. So in previous years, only solvent extractions were preferred, especially chloroform and methanol. But now aqueous extracts are also being increased, and a very good pharmacological activities are being reported. So probably the R&D cost can be reduced finally by applying all the green chemistry concept at every level of synthesis. So obviously every company wants to make business. So if green chemistry is utilized and implemented properly, I think the company's business is going to sustain for few more years, or else probably it has to work out in the future. So each company has got its own strategy to reduce the toxicity level or hazards in the individual sectors. So I could find one parameter in order to find out the change in the level of toxicity, that is Rowan solvent greenness scoring index. So this is one of the parameter to assess the reduction of toxicity of the solvent. So in this, if we see for in the beginning, for preparation of a compound, they have used process 1 using 4 solvents, each 25 kgs. So the greenness index was found to be 419. So this is pretty well very high. Over a period of time, by applying the concepts of green chemistry, they could improvise the synthesis of the same compound by eliminating one of the solvent. So 25% of the solvent usage is reduced, and in that process too, the greenness index has been reduced to 285 from 419. So this parameter calculation itself is an indication that every process can be streamlined to bring down the hazardous property of the process. So you have the greenness scores for calculating various solvents with regard to environmental waste, environmental impact, health and safety. So there are lots of measures and parameters or yardsticks that are available in the internet and even in lots of publications, where an individual firm can establish their own standards, because whatever I am showing it is not a gold standard. It is only an example that what people are doing in order to arrive at a safer level of preparation. So you can work out with all the solvents which you are using daily from morning to evening in every process, and you need to work out how the percentage of solvents can be reduced or the usage pattern can be improvised. On these processes, exercise can be done. This is another example. Sildenafil is one compound where again solvent was used. So now, right from its discovery, if you see, initially for synthesizing they used 1540 kgs of this per kg of sildenafil. Now there was a process improvement again made, and the amount from 1540 kg was brought down to 94 kgs. So this is almost an excellent reduction. Then you can see a gradual decrease, and finally in the end the future target is they are going to use only 4 kgs of this solvent per kg of API. So probably I think this could be an excellent improvement in order to reduce the overall consumption of the solvent. So indirectly the cost cutting is made, at the same time safety measures are improved. This is another example, waste generation per kg of sitagliptin produced. Initially the total percentage, if you see, is more, and after the new route is synthesized, almost 60% of wastage is reduced. So in the principles of green chemistry, I have shown the first point, prevention. So if you reanalyze the entire process, if you can prevent the wastage, indirectly again there is a cost cutting factor. And this is another example, dichloromethane used at small molecule discovery sites. So because many of the times chloroform, dichloromethane, methanol, they are being used for solubilization and analysis. So here again we can see here from the DCM usage is being reduced from 120.4 to 51.9. So I have collected some of the cases of some pharma companies only to show you that a great paradigm can be made in the structure of pharma company processes or any other processes. So the importance of green chemistry is also in terms of nanotechnology. So this is, I have only shown you at the synthesis level. When we talk about nanotechnology, I think nano products are gaining more and more important because they have got a specific delivery properties to deliver the drug at a proper rate to the site of action. Many of the nano systems also use tremendous use of solvents. So green chemistry concept now is increasing even at the level of nanotechnology. So in recent years, the development of efficient green chemistry methods for synthesis of nanoparticles has become a major focus for many of the researchers. Because we can see thousands and thousands of papers are being published every month only in the area of nanoparticles. So probably I think we can look into the aspect of green chemistry even in the area of nanotechnology. So an eco-friendly technique for the production of well-characterized nanoparticles can be made. That means an alternate synthesis can be selected, and production of metallic nanoparticles using organisms. Using microorganisms also, nanoparticles can be synthesized by substituting or by removing the usage of solvents. Similarly, plants have become one of the best sources for preparation of nanoparticles. Plants contain substances which are more safe than drugs which are synthesized by the process of chemical synthesis. So nanoparticles produced by plants are more stable and the rate of synthesis is faster than in the case of microorganisms. So either the microbes can be selected or plants can be selected so that the number of steps are reduced, rate of synthesis is increased, toxicity is reduced, cost is reduced. So if we look into the life cycle of nanoparticles, you have material processing. Because to prepare any nanosystem, I require 5 to 6 ingredients. Each ingredient, if I want to supply in nano size, then probably the point of processing of materials comes into picture. So if the processing again is not considered in the with regard to hazards level, then probably the occupational exposure for that material will be high, as I've mentioned now, and that can lead to toxicity. And in manufacture, if you are going to use high energy production methods, then also it is going to be very toxic to the person who is going to handle. So coming to the usage, the exposure to humans in the environment. Now I have mentioned just now, a person was complaining about loss of hair by handling anti-cancer drug because of exposure. So that exposure need not be only an anti-cancer drug. It can be a solvent or it can be any other chemical in the near the working environment. So such measures should be made while preparing nanoparticles whenever solvents are used. And ultimately, normally while preparing nanoparticles, 25 to 40% of the solvents are recovered to make the process more economical. So while recovering, if any toxic products are again coming into the solvent, and if that cannot be eliminated, that is going to become a major problem in the coming next synthesis of the nanoparticles. So in the literature, we have umpteen number of nanoparticle synthesis methods. So here, classification wise, one is bottom-up approach. Bottom-up approach means you are going from molecular level to particulate level. And this is top-down approach, that is from particulate level to molecular level. So in bottom-up approach, especially if you consider the method of templates. Now this is one method where the amount of solvent used is very high. In order to solubilize, it may be surfactants, it may be lipids, it may be drug or anything which you are going to use in the preparation of nanoparticles. So in this case, the presence of residual amount chances is very high. Therefore, I cannot say that the product is 100% going to be toxic free. At the same time, if you look into the top-down approach, like for example, mechanic milling, the technology in this milling process, now you have a high pressure homogenizer, ultrasonicator. Now these equipments are excellent enough to bring down the size of the particles at nano level, like 100 nanometers. But the amount of energy that is going to generate is tremendously high. So if this energy disturbs the system, like if this energy it tries to initiate degradation mechanism in the particles, then again you cannot select this process. So considering green chemistry at the level of synthesis of nanoparticles, the alternative approaches are either you can go for usage of microorganisms because they can degrade or they can process the complex materials into simpler materials, thereby obtaining the nanoparticles, or directly plant extracts can be selected for synthesizing the nanoparticles. So you have metallic nanoparticles being coming up. Metallic nanoparticles are excellent nanoparticles mainly used in antibiotic resistance. These metallic salts, they bind to DNA, prevent DNA replication. At the same time, they increase the oxidative stress in the microorganism. So, but the problem is when metallic salts are given as such, they do not have the specificity. So when it is incorporated in the nanoparticles, the specificity is given more. So this is an illustration of preparation of nanoparticles using green chemistry. So here, instead of solvent, simply they have used alkali. So here, by just altering the pH, the size was optimized to 100 nanometers, and that's how they could prepare a targeted metallic nanoparticle to treat infections of interest. And surface modification is another important property in green chemistry in the preparation of nanoparticles. So people can use surfactants to increase the solubilization, because solubility is going to be a major problem in many of the drugs. So that solubilization can be manipulated at the level of size fraction or self-assembly. So further, this property can be enhanced by modification of the surfactants. So all these are some of the pictures of metallic nanoparticles where the total removal of the solvent has been made, only water is being used with alkali pH, and you can see there is no change in the efficiency in the production of nanoparticles in spite of removal of the solvent. So that itself is an indication that yes, we have lots of other alternative methods even in the preparation of nanoparticles where solvent amount can be reduced. This is again another picture of preparing magnetic nanoparticles. Even to nanoparticles, magnetic property can be incorporated so that targeting property can be incorporated. So this is the separation of magnetic nanoparticles by using magnetic field. And the applications of magnetic nanoparticles is also increasing, especially in pharmacology, toxicology, and pharmaceutical applications. So finally, the use of green chemistry in the pharmaceutical industry with regard to the safety, environmental toxicity, cost is going to play a major role for many of the pharmaceutical industries and non-pharmaceutical industries. So let us hope that the design of new upcoming pharma industries take into consideration the concept of green chemistry so that the working environment can be made more safer and at lesser price without compromising on the final quality of the product. So the conclusion is that with the unique properties of green chemistry, especially in pharma sector, non-toxic manufacture of metallic nanoparticles can be made so that treatment modality can be improved, solvent consumption can be reduced to a drastic level, a safer environment can be provided to the entire sector with minimum cost. So with this, I would like to thank you one and all for your kind attention and patient listening. Thank you.
Thank you, Professor Kumar. Time for one question.
Okay, thank you for your talk. Yes, it's fine that you encourage us about the role of green chemistry in pharmaceutical industry. But actually, I have a comment that you didn't talk about the pharmaceutical analysis. This is a big sector also in industry, pharmaceutical industry, regarding the quality control and so on. So this is also consume a lot of organic solvents, and you need to modify a lot of methods in this sector. I talked about this yesterday for the using of capillary electrophoresis instead of HPLC. Okay, I have another question that you are talking about greenness factor. You know any standard limit, international standard limit for this factor?
I don't have, but actually many of my friends are working in pharma industry at managerial level. So they are planning to start a new pharma industry. So what they have told is, now as per the new terms and conditions, to provide a safer environment, what kind of steps can be taken at the level of construction, and that have to be discussed with the engineers. So in that intention, I made a point on that. And regarding, as you already told, that analysis, I made a point on HPLC consumption because of.
Okay, thank you very much for your nice talk, and we continue with the next speaker.
Yeah, thank you.
Thank you. Our next speaker is Professor Puong from Vietnam, who is going to speak about chromate and arsenate removal by layer double hydroxy polymer beds.
So, good afternoon, everyone. My name is Phượng, Nguyễn Thị Kim Phượng. I come from Vietnam. So why I choose this? Why I work with the chromium and arsenic? Because as you know, our country have big problem with the contaminated, especially the groundwater. The people who live in the Mekong Delta, the groundwater is contaminated highly. High concentration of the arsenic, sometimes is 10 to 100 times compared to the acceptable limit. And chromium is come from the industrial activity. That's why we work with this. And also the aim of this research is that we are looking for the cheap material to remove the chromium and arsenic. And it's also easy to apply. So as we know, the layer double hydroxide is the anionic clay, and it's used for many purposes before, like for medicine, for the catalyst, and environment remediation. And due to the high specific surface area and high anion exchange capacity and flexible layer space, so it's good candidate to remove the negatively charged species. And however, it has the problem is that the use of the LDS in the powder form is have the big problem for the after treatment. That is the separation between the liquid and solid. So if it's easy to remove from the our media, so LDS will be the most potential candidate. And so far, various forms of the layer double hydroxide is have been developed, and some researchers before previous is also try to coat it in sand or zeolite or on the cellulose. And recently, we heard more about the entrap, the functional material in the biopolymer, especially is in the alginate. in our group is also work on that, and actually it's before previous study we entrap the layer double hydroxide in the anginate bead only, but it's easy to break down after one cycle using, so we modify, slowly we modify and we put it with the Bolivian alcohol. Also we use the cross-linker aldehyde, so it makes the bead become stronger, so we can recycle for several times. So now I will introduce about how to prepare the layer double hydroxide. That is a very easy way to prepare by co-precipitation methods in the very, how to say, very high BS, at BS 11. And I use two precursors. One is the magnesium and aluminum, and another couple is the magnesium with ferric. And after we do precipitation, so we can age in a 65 Celsius degree for 24 hours. And after separation, we heat it to 450 Celsius degree for 4 hours. So it's ready for prepare the bead. And also from the previous study, so we find out the bead with 8% of the layer double hydroxide. 8% is a very good absorption. So after we prepare the bead, we let it cool 24 hours in the calcium chloride solution. And to make sure that here is our material, the white one is the magnesium, aluminum layer double hydroxide, and the red one is magnesium ferric layer double hydroxide, and this one is a blank one, only polymer. And to make sure that our material we prepare is correct, so we use ICRD to confirm, and it's, our result indicate that we prepare the correct layer double hydroxide. And we use the SEAM and ICRD to see whether the layer double hydroxide is mixed well with the polymer, and from the result we can find out that our material is a very good disperse. So it's ready for the experiment. One is lost. So we use the material for the to remove the chromium and arsenic. Actually, this is just very basic research, and we find out that the for chromium is the optimum BS to remove 7 and around 90 to 92% of the chromium were removed by our material. And for arsenic is at BS7 is the optimum condition, and the removal efficiency is only 79 to 91. We also test the reuse the material, and we find out is that our materials can be stable at least 5 cycle absorption desorption cycle, and the absorption ability is just reduced 5 to 6% at the 5th cycle. And we also use some to understand the mechanism, absorption mechanism. So we use the pseudo first and second kinetic to fix our data, and we find out is that our data is good with the fit well with the second kinetic model. Also, we use the Langmuir and Freundlich isotherm to fix our result, and we find out our result is good with the fit well with the Langmuir. And actually, is the best but absorption just for to understand the reaction between the pollutant and absorbent only, and it also gives us help to understand to get the best operation condition. However, if you want to apply for the real for the big scale, so another mode will introduce for using, and the feedback column is the best way to apply for the remove the pollutant. And here is we run in the column. So we use the upflow mode, and the bed depth is 40 centimeter. The flow rate is only 3 milliliter per minute, 3 ml only. That is mixed. And we also, after run the experiment, we also use the kinetic model to fix our experiment. And here's our result. And we find out that our result is also fit with the Adam Bohr's kinetic model, and it's fit good. And the bed depth we back with the magnesium aluminum bed is service longer time and larger volume treatment, and it's longer service. And based on the results, we also calculate some kinetic data. And we also find out the total removed by our column is from 86 to 91 for chromat, and for arsenate 89 to 90%. Okay. Conclusion is that our material is satisfy the needs of the cost effective, and it can reuse for many, at least 5 cycles. And the removal efficiency is 90 to 92% for chromium and 79 to 91 for arsenic. And we can the absorption mechanism is follow the Basedow second other kinetics and fit well with the Langmuir isotherm. In the column, we find out that the breakthrough time is 10 to 15 for chromium and 6 to 8 for arsenic. And this result can also use for the further research, and it will be the promising absorbent. But actually, right now in my lab, if we start to work with the real live water. So for arsenic, we use the groundwater from Mekong Delta, and for the arsenic, we use the wastewater from the industry for like 10, like the metal plate, something like that. Yeah. Thank you.
Thank you for the nice talk. Some questions? I expect it.
Okay, that's nice to remove hexavalent chromium, but you studied the removal of hexavalent chromium in the presence of trivalent chromium, because you know the reduction from hexavalent to trivalent is easy.
You mean you make the reduction from the chromium, hexachromium to the chromium 3. Actually, if you go to that step, so you will use a lot of the chemical, and use a lot of lamp. The hydroxide, you how to say, precipitate the chromium 3. So before the many years before, for treatment the hexachromium, the people have to use the some chemical to reduce, and after that have to precipitate. So it's create more sludge. So problem is you have to zone the sludge.
So you mean you should precipitate
So absorption is the best one. Absorption is the best one.
Oh.
Yeah.
Okay. Thank you.
More questions? It's not the case. Let's thank not only to the speaker, but also the speaker of the afternoon, because we are going to change the session. We are going to continue with the June Research Forum. Yeah, but he said later on, no? You want to give now? Okay. So, the certificate for Professor Kumar. He left? No, there are only one Kumar. So Andrea Cotwain is not here? No. But we give before this, okay. For the young researcher, Juliana Velázquez is here? No? And Dominic Gotts? Okay, please. Come in. They want to give first the certificate, so congratulations. Katarina Fabikovica. Here. Okay. So congratulations. Thank you. And, well, Professor Nguyen, your certificate. Congratulations. That's all? So we can start with the forum? Okay. So the next speaker is Katarina Fabikovica, but I think she's not here.
I'm here.
Yes. I always sorry. So you can start with your talk.
Okay, thank you very much. Good afternoon, ladies and gentlemen.